In 1971, Gunther Blubel and his colleague David Sabatini proposed secretory proteins might carry a short segment that directs their trafficking through the intracellular environment. Blubel and Bernard Doberstein successfully proved this hypothesis in 1975 using canine pancreatic cells. This discovery led to Blubel winning the Nobel Prize for Physiology or Medicine in 1999.
The major and minor characters in our show today are as follows. mRNA is a nucleic acid sequence which codes a future protein. Ribosomes are the cellular machinery that facilitate production of proteins. SRP stands for Signal Recognition Particle, which recognizes a hydrophobic region of a protein. It halts protein synthesis until complex docks at SRP receptor in ER membrane. The ER is a membrane-enclosed organelle that assists in membrane protein translation and localization. Lastly, chaperones and calnexin assist in the folding of immature proteins.
An mRNA and a ribosome meet and fall in love. They decide to make a protein. As it begins to grow, it becomes apparent that something about this protein is special. One day, a shadowy agent emerges, and the mRNA and ribosome discover that their protein is not just special, it is chosen. The SRP informs the protein that it has a stretch of nonpolar amino acids. Bum bum bum! Now that this protein's destiny is apparent, the SRP and its parents must depart on a journey to a place where it can be among its kind.
The SRP docks on the SRP receptor helipad. The young protein is loaded into the protein translocator and finds that it must journey on alone. The protein descends into the ER, leaving its parents and chaperone behind. As the young protein descends into the depths, the SRP refuels and flies off in search of the next gifted youngster.
Once the translocator binds the hydrophobic sequence, the ribosome continues translating mRNA. Upon completion of translation, this mature protein is released into the ER lumen and a signal peptidase cleaves the initial hydrophobic sequence. Chaperone proteins facilitate post-translational modifications to the mature protein within the ER.
Mic check, mic check, mic check, mic check, mic check, mic check, mic check.
The protein is now in the ER in an unfolded state. It has a N-linked oligosaccharide with 3 glucose molecules. 2 of these molecules are then trimmed off to allow the protein to properly bind to calnexin. Calnexin then folds the protein and removes the last glucose residue. At this state, the protein is either properly or improperly folded. If improperly folded, glucosyltransferase adds a glucose residue back onto the protein so that calnexin can try again. This process will continue until properly folded, and now the protein can leave the ER.
Proteins are transported via COP2-mediated vesicles from the ER to the cis-Golgi network. Proteins with a signal sequence known as KDEL, corresponding to 4 specific amino acids, are recognized by a KDEL receptor and transported back to the ER via COP1-mediated vesicles. These proteins containing the KDEL sequence are referred to as missorted ER proteins.
Localizing proteins to the ER is just the first step of localizing any proteins found in membranes or in vesicles. Within the cell, the process of localization to the ER is responsible for all proteins that end up in vesicles, like the enzymes and lysosomes, and in membranes, like the receptors bound in the cell membrane. So without the ER, cells would not be able to take up any material or break down any waste products, among a lot of other things. In short, without the magical endoplasmic reticulum school for gifted proteins, you, or any organism, would be very, very dead.
I'm picking up good vibrations. She's giving me the excitations. I'm picking up